Hi, my name is Isaku Tanita at Element Biosciences. As part of a series of short videos highlighting the value of the AVITI system for single-cell analysis, today I'm going to show you a recent study we completed with our partners at BD Biosciences featuring the AVITI system working seamlessly in combination with two of their BD Rhapsody single-cell assays for characterizing human immune response. First, you may recall from our earlier video on single-cell analysis, when you set up these types of experiments along with the sequencing that's required for them, you need to account for several important criteria. You need flexibility to QC your libraries, like sequencing a subset of samples before you sequence your entire project, or to pool libraries together, or to accommodate different single-cell assay types, which are growing in variety. And then there's throughput. More samples or more cells or more reads per cell, it adds up quickly as your project evolves. High quality plays a part as well, and much like the sample quality itself, the sequencing quality also needs to be high to maximize the biological insights that you can gain from your samples. And of course, cost and accessibility play a large part in the process where users seek to minimize expense and delay. All told, the AVITI system provides an ideal solution to meet these needs with performance features that make single-cell analysis by NGS more accessible to labs than ever before. In our single-cell case study today, using the BD Rhapsody single-cell analysis system, you can see how the AVITI system fits within the workflow in this diagram. The Rhapsody system begins with a proprietary microwell-based partitioning technology for capturing and barcoding thousands of single cells. These cells are ready to use within a menu of proprietary BD single-cell assays. To demonstrate platform compatibility between the Rhapsody system and the AVITI system, mRNA and surface protein markers were isolated by loading 10,000 and 2,000 ABSEQ-stained human PBMCs onto a BD Rhapsody cartridge using the BD Rhapsody system and enhanced cartridge reagent kit. Following cDNA synthesis, WTA, BD ABSEQ Immune Discovery Panel, and Targeted Immune Response Panel libraries were made compatible for sequencing on the AVITI system using the Element Adept Compatibility Kit, a simple 75-minute protocol in which libraries are circularized without the use of PCR amplification. After sequencing on the AVITI system, the AVITI generated FASTQ files which were analyzed with BD GeneSeq software. Using the WTA, ABSEQ, and Targeted Human Immune Response Panel libraries created as replicates at the two different cell counts, you can see the strong respective performance metrics tabulated here. By scanning across the three rows indicated, you can see the high sequencing quality of the AVITI system indicated by the high percent Q30 from 93% to 95%, the high percent reads assigned to cell labels from 96% to 100%, and the low percent reads filtered out. And within the pairs of columns, you can see internal consistency within the library types between the two starting sample sizes for each library. In this slide, we further compare results in R-squared and t-SNE plots for the respective libraries. Panel A on the left side shows a high gene expression correlation between the high and low cell input WTA plus ABSEQ libraries. t-SNE plots from targeted libraries in Panel B illustrate the distinct clustering of PBMCs by cell type with no batch effect between high and low cell input. Panel A on the right side shows similarly high gene expression correlation between the high and low cell input targeted mRNA HRRP libraries, and Panel B similarly shows t-SNE plot clustering consistent with no batch effect between high and low cell input. And before we summarize, let's step back and take a quick look at what all this can mean in terms of sequencing costs on a per sample basis. This table reminds us of the tremendous cost savings and flexibility the AVITI system offers for single-cell analysis compared to alternative benchtop systems, both at the 2x150 read length and especially if you're using the shorter read length assays at 2x75 or less than 150 cycles. I hope this review was helpful to understand how easily the AVITI can plug into the BD Rhapsody system for single-cell analysis, and to remind you of the tremendous user advantages and sequencing cost savings the AVITI can mean to you when you're planning your next experiments. A thank you to our partners at BD Biosciences for their collaboration and support, and for more details on the performance of the AVITI with the BD Rhapsody system, please feel free to download our technical note on this subject from the Element Biosciences website. And most of all, don't forget to look for the upcoming release of our 2x75 sequencing kit, offering the lowest paired-end sequencing for single cell in the industry. Thank you.